The planning of arm-reaching movements is achieved through a cascade of sensory motor actions, from localizing and determining the goal to generating the motor commands to complete that goal. These motor goals are encoded in multiple reference frames. The majority of previous research in this area has been performed using monkeys as the subject. A set of areas in the dorsocaudal posterior parietal cortex, or PPC, are thought to be specifically involved in the planning of arm-reaching movements because the majority of neurons in this area are more active during the delay period for instructed arm movements than eye movements. This area is referred to as the parietal reach region. Neuronal activity in this region best describes the direction of subsequent arm movements using a gaze-centered reference frame, even when the targets are non-visual auditory targets. These findings argue for a key role in the dorsocaudal PPC in providing an early high-level description of the motor goal in extrinsic visual coordinates, which can then be transformed into intrinsic proprioceptive coordinates. Proprioceptive refers to a stimuli that is produced and perceived within an organism. Electrophysiological recordings in monkeys indicate that the PPC is an important area for the computation of goal-directed actions and that it contains specialized regions for different target locations. But can we generalize the finding of monkeys to human brain functions? Maybe. There's growing evidence suggesting plausible functional homologies between the human and monkey PPC. In humans, several neural imaging studies have identified a region in the dorsomedial PPC that is also more active during the pre-movement preparation for arm movements than for visual saccades. As in the monkey, neuronal activity in the human PPC represents visual target locations primarily in gaze-centered coordinates. Also, reach errors associated with optic ataxia, which is a sensory motor deficit arising from PPC lesions, depends on the dynamic gaze-centered internal representations of remembered visual targets. Despite considerable evidence for a predominant role of gaze-centered information for visual reaching tasks in both human and monkey PPC, it remains to be shown whether a visual representation is also used when goals are solely defined by unseen proprioceptive signals. It was unknown whether proprioceptive cues elicit the flexibility that other studies have found when weighing the extrinsic and intrinsic representations of a given motor goal. A long-standing challenge in this field has been to determine where these representations are functionally nested in the brain. Bernier and Grafton hypothesized that the reach-related regions of the human PPC may demonstrate a capacity to use different frames of reference as a function of the sensory modality of the target. To address this question, they manipulated the sensory nature of the target for reaching, either visual or proprioceptive. They use fMRI repetition suppression to directly observe which reference frame the motor goal is defined in. In this study, there were 18 right-handed participants from the University of California, Santa Barbara. All subjects had normal vision and had no history of neurological diseases. Participants are positioned supine in the scanner with head and neck cushioned to prevent movement and wore headphones to receive auditory stimuli. Visual stimuli were presented on a fiberglass board resting on the participant's abdomen, and the participants utilized a mirror above them to view the task area. These tasks were done in darkness, and so at no time could participants see visual feedback of their hands while performing the tasks. When participants press a button, a central fixation point appears at the center of the board. Participants are then instructed to fixate their gaze at one of 4 points, either 15 or 5 centimeters to the left or the right at the central fixation point, ensuring that tasks are performed in the peripheral visual field. After 2, 4, 6, or 8 seconds, participants received an auditory instruction to make a reaching movement to one of 2 red dots that appear 5 centimeters to the left or right of the central fixation point. The proprioceptive task is conducted in the same fashion, except participants reach towards the unseen left thumb and left index finger, which are now taped behind the fiberglass board shown in this image. The positioning approximates the positioning of the red lights in the visual task. 2 aspects of the visual and proprioceptive task were measured: gaze-centered target position and body-centered target position. In this condition, the target is right with respect to gaze and right with respect to body. In this case, the target is right with respect to gaze, but left with respect to body. The 2 target conditions were performed in separate sessions 1 day apart. Each session comprised of 8 runs. In 4, participants performed the reaching task, and in the other 4 runs, they performed static tasks. The amplitude of the reaches were short, about 10 centimeters, so they could be largely performed with the wrist, minimizing arm movement. The researchers explained that as there is still displacement of the arm, this constitutes reaching as opposed to pointing. Brain images were temporally high-pass figured at 128 seconds cutoff and smooth with an 8mm full-width half-maximum Gaussian kernel. Individual-level fMRI analysis was performed using the GLM, addressing target modality and the suppression of the bold response. Second-level random effects analysis were applied to each individual contrast to obtain a population estimate. Researchers employed a rapid, single event-related design using a 2x2 factorial matrix with reference frame, either gaze or body, and target modality, either visual or proprioceptive. A region of interest approach was used to assess the relative repetition suppression, and the ROIs were defined as 8mm spheres around the peak of each. A suppression index for each participant was calculated using the differences in mean parameter estimates between repeated and novel trials. These suppression indices were analyzed using a two-way repeated measures ANOVA.
The first objective was to find the brain areas associated with reaching movements to both visual and proprioceptive targets. This was done by comparing activations seen in both the reaching tasks to those seen in a static task where no reaching was done. Overall, a similar set of reach-related regions was observed for both visual and proprioceptive tasks, leading to widespread activation in largely overlapping regions of the parietal frontal network. In both the visual and proprioceptive tasks, reaching to the left with respect to both body and gaze created greater activation. It is thought that this is related to the greater effort associated with moving the right hand to the left. Next, they wanted to test their hypothesis by determining which of these reach-related brain regions of the parietal frontal network was associated with gaze-centered and/or body-centered coordinates in both the visual and proprioceptive tasks. Essentially, does this pattern depend on the sensory modality of the target? Areas that showed sensitivity to repetition suppression during the tasks were associated with that motor goal and modality of the target. Overall, the main areas involved in both the visual and proprioceptive reaching tasks were the anterior precuneus, area BA5, PMD, PMV, and POJ. However, the most important finding was that the use of reference frames for action according to sensory context is not achieved by engaging different brain areas when using either gaze or body-centered reference frame. It is done by recruiting many of the same areas that themselves change their mode of representation, either gaze or body, depending on whether the sensory modality is visual or proprioceptive. An ROI analysis revealed that all areas activated in the visual target condition showed some level of sensitivity to gaze. In the proprioceptive condition, the same areas actually reversed their mode of representation to show strong repetition suppression for body instead. As you can see in the bar graph, the anterior precuneus predominantly showed repetition suppression for gaze in the visual target condition, but the same area reversed its reference frame to body-centered in the proprioceptive condition. The ROI analysis also revealed a pattern of repetition suppression in brain areas showing a mixed representation, meaning they represented both body and gaze reference frames in the same area. BA5 and PMD were two brain regions that showed these patterns. As you can see in the bar graph, repetition suppression for gaze and body didn't differ in the visual condition, but repetition suppression for body was significantly greater than repetition suppression for gaze in the proprioceptive condition. According to this view, the brain simultaneously maintains gaze and body-centered representations of the motor goal. However, the relative contribution of each of these reference frames to the output would be dependent on the available sensory cues.
Previously, it was shown that the PPC strongly represents the visual inputs of a motor goal, but it was uncertain if the area also uses a visual code for movements that are directed towards proprioceptive targets. The study provides evidence for a heterogeneity of representations for visual reaching, given that the same area was activated in body-centered encoding and in gaze-centered encoding. This insinuates that the areas recruited can change the mode of representation rather than the idea that flexibility means recruiting different brain regions for different tasks. This study also supports other work in the precuneus relating it functionally to the PRR in monkeys, where goal-oriented activity is specific to arm reaching. The present data about gaze-centered encoding of visual targets agrees with that view. It was previously thought that there was a single area in the PPC that would receive all input, whether body-centered or gaze-centered, and simplify them by remapping them to gaze-centered representations. That now appears to be too simple, and instead there does not appear to be a rigid assignment of coordinate frames to a specific area of the cortex. Research in monkeys indicates that their PEC area receives extensive projections and is modulated by passive and active upper limb movements. Given this, it may make more sense to think of the PPC as containing neuronal populations capable of flexibly representing the motor goals using mixed reference frames. There are several areas for improvement in the methods of this study. First, in the static task, there was no attentional instruction. Attention is thought to engage the anterior precuneus, so it is unclear whether this affected the results of the study. Second, it is not possible to determine the reference point for the body-centered representations, and through the bold response, you can't detect what the anchor point is. This information would be relevant in the context of future studies. The study has had a lot of variables in its event-related design, which led to widespread activation for a variety of reasons. It might be beneficial to tease out these functions individually and be more specific in the goal of the study. Only right-handed participants were involved, and some of the activated areas were biased to the left side of the brain. The addition of left-handed participants would ensure that this information is relevant. Another shortcoming in the conclusions drawn is that much of the work in monkeys was done through single-cell recordings, and drawing parallels between that data and the fMRI data from this study is a bit of a stretch. The most exciting avenue for future research is the possibility of neural prosthetics. Prior to this being possible, further elucidation of neural mechanisms would be required to better understand how movement is encoded in the PPC. One way to do this would be through single-cell recordings in monkeys. The present study provides a new means for assessing coordinate reference frames for actions in humans. For neural prosthetics to be more applicable to humans, it would have to be made to be less invasive and confirmed further in animal models, but is nonetheless an exciting prospect.